channel. Today in the fast track revision series we will be learning about enzyme inhibition. So let's see what are inhibitors. Inhibitors are the substances which decreases the rate of an enzyme catalyzed reaction causing temporary or permanent stoppage of the enzyme activity. And based on their binding with the enzyme, inhibitors are of two types: irreversible inhibitors and reversible inhibitors. So first we will look at irreversible inhibitors. Irreversible inhibitors bind covalently to the enzyme, means they bind strongly to the enzyme and either dissociate very slowly or does not dissociate at all, leading to the destruction of the functional groups in the active site of the enzyme, and hence enzyme's activity is lost. And most of these inhibitors are toxic substances. So as the name suggests, irreversible inhibitors, once they bind to the enzyme's activity can't be reversed. So let's see some of the examples of the irreversible inhibitors. The first example is of diisopropyl phosphofluoridate, DIPF. DIPF binds to the serine residues in the active site of acetylcholine esterase. And if you remember, acetylcholine esterase is that enzyme which converts acetylcholine into acetate plus choline, present in the synaptic cleft. So when DIPF binds to the acetylcholine esterase, this enzyme is no longer functional, and this will lead to the accumulation of acetylcholine in the synaptic cleft. So when acetylcholine would accumulate in the synaptic cleft, it will lead to unnecessary stimulations and leading to a condition referred as neurotoxicity. The next example is of parathion. Parathion is an insecticide like DIPF, but it is specific for the insect acetylcholine esterase. Third one is physostigmine and serine. Serine is a nerve gas. It is also like DIPF. And fourth example is of penicillin. Penicillin inhibits the enzymes that are responsible for the bacterial cell wall synthesis in irreversible manner. So let's go to the next slide. And now we will look at the reversible inhibitors. Reversible inhibitors. Reversible inhibitors bind non-covalently with the enzyme, means they bind loosely with the enzyme and dissociates rapidly. So on dilution of enzyme inhibitor complex, enzyme's activity can be regained. So what is the meaning of dilution of enzyme inhibitor complex? It means that when we add more and more substrate, it will lead to the dilution of the enzyme inhibitor complex. This substrate would replace all the inhibitors from the active site of the enzyme and will capture the active site of the enzyme. So enzyme's activity can be regained. And there are three common types of reversible inhibition: competitive, non-competitive and uncompetitive, and the first two are important for the NEET aspirants. So let's go to the next slide, and first we will look at the competitive inhibition. So as the name suggests, these inhibitors compete with the substrate for binding with the active site of the enzyme and is structural analog of the substrate. Means if anything wants to compete with the substrate, then it has to look like the substrate. So same thing happens here. If you will look at this equation here, E plus S is enzyme substrate complex, and it is finally forming enzyme plus product. But at the initial stage, this substrate is stopped by inhibitor from binding with the active site of the enzyme. So the inhibitor forms an enzyme inhibitor complex that is equivalent to the enzyme substrate complex. And as we have already seen that on dilution of the enzyme inhibitor complex, the enzyme's activity can be regained. Now let's look at this curve. So if you remember Michaelis-Menten equation, then this curve is not really tough for you. On the x-axis you can see substrate concentration, on the y-axis velocity is there. Vmax is the maximum velocity that can be achieved, and Vmax by 2 is the half of the maximum velocity. And what is Km? Km is the substrate concentration at which reaction reaches half of its maximum velocity. Means the concentration of substrate at which reaction reaches half of its maximum velocity. So that is Km. And Km tells you about the affinity of the substrate for the enzyme. Means it tells about the love of the substrate for the enzyme. If the value of Km is high, then the affinity of the substrate for the enzyme would decrease. And how that happens, let's see. What we are doing in competitive inhibition, suppose I am representing this as N, means this is a normal situation, and this as Ci, we are adding the competitive inhibitor here. So in normal situation the Km is this, and once we add the competitive inhibitor, the Km increases to this. Means we have to add this much of substrate to achieve the new Km, although the Vmax is at the same place, but we have to provide extra substrate. This means with increase in the value of Km, the substrate's affinity for the enzyme is decreasing. So that is what is seen in the competitive inhibition. In competitive inhibition, on increasing the substrate, Vmax can be achieved. So Vmax remain unchanged while the value of Km increases. Now let's look at another plot. This is called as Lineweaver-Burke plot, and it is obtained by taking reciprocal of the Michaelis-Menten equation. So from this plot also it is very clear that Vmax is same for both normal and competitive inhibitor and normal case. As you can see, this is Ci and this is no inhibitor case. So the Vmax is same, but you can clearly see that the Km is increasing. That means in order to achieve the same Vmax, we have to provide more substrate. Now let's look at some of the examples of the competitive inhibitor. Before that, we'll see two statements that are very important. The effect of competitive inhibitor on activity is reversed by increasing the concentration of substrate. We have seen it, this earlier. And as at high substrate concentration, all the active sites of the enzyme get occupied by the substrate, and reaction velocity reaches the value observed without an inhibitor. Means as we increase the substrate concentration, this substrate replaces all the inhibitor from the active site of the enzyme and itself get captured inside the active site. So that's how the enzyme's activity can be regained. Now these are, if you remember, this is the Michaelis-Menten equation, and this is the value of alpha. Alpha is basically denoting 1 plus concentration of inhibitor, and Ki is the inhibitor dissociation constant. And if we take the double reciprocal form of this equation, something like this would be obtained. So NEET aspirants could skip this part. Let's go to the next slide for the examples of the competitive inhibition. The first and the very important example of competitive inhibition is of succinate dehydrogenase. This enzyme is a very important enzyme as it is required in both citric acid cycle as well as electron transport chain. And what is the function of this enzyme? This enzyme catalyzes the conversion of succinate. As you can see, this is the structure of succinate, CH3, CO minus, CO minus, CH3 to fumarate. And if you want to stop this conversion from succinate to fumarate, you need to add a malonate. Malonate is a structural analog. It looks similar to the succinate. So this is how we control the growth of bacterial pathogens. In case of bacteria, if succinate's conversion to fumarate can be stopped, then their growth can be stopped. Let's move to the next slide for another example. What happens in case of alcohol poisoning? So as you know that methanol is used to adulterate the ethanol. So when a person suffers from alcohol poisoning, means there is very large amount of methanol that he has consumed, and this methanol gets converted into formaldehyde by the enzyme alcohol dehydrogenase, which is present in the liver. And this formaldehyde is very toxic for the cells, as it is converted into formic acid, leading to the metabolic acidosis. Means the pH of the cells go very down, due to which the cells die. And in order to prevent this, doctors give high doses of ethanol, which is a competitive inhibitor of methanol. It competitively inhibits methanol and replaces methanol from the active site of ADH, preventing the formation of formaldehyde. So that's how you treat alcohol poisoning patients. Let's see the next example. This example is of control of bacterial pathogens by sulfa drugs. So let's see what is happening here. The first structure is of para-aminobenzoic acid. So as you can see, this is ortho, meta and para. So on the para position you can see there is an amine group. So this structure is called as para-aminobenzoic acid, and this substrate is very important for the formation of folic acid. Folic acid is necessary for the growth of bacterial cells. So the enzyme which converts this para-aminobenzoic acid to folic acid, we can add a competitive inhibitor of para-aminobenzoic acid, and this competitive inhibitor is sulfanilamide, or there is a group of these competitive inhibitors, and they are collectively referred as sulfa drugs. So these drugs, they look very similar to the para-aminobenzoic acid. As you can see here, there is amine group on the para position and SO2 and on the first position. So this looks very similar to the PABA, and it competitively inhibits the PABA. Now we will study about non-competitive inhibition. Non-competitive inhibition, first look at what are non-competitive inhibitors. These inhibitors does not share any structural similarity with the substrate and binds to other site than the active site. Means non-competitive inhibitors, suppose this is an enzyme and this is an inhibitor and this is a substrate. So substrate would bind to the active site of the enzyme, while this inhibitor will bind to some other site of the enzyme. So when this inhibitor is binding to some other site of the enzyme, suppose I am showing inhibitor here, so the active site is no longer required by the inhibitor. Then due to binding of inhibitor, enzyme's 3D configuration is altered and the reaction is blocked. So inhibitor either binds to the free enzyme or the enzyme inhibitor complex. We'll see the reaction in the next slide. Since the active site is not targeted, no matter how much substrate you provide, the reaction won't get reversed. Means once the inhibitor has bound to some other site of the enzyme, no matter how much substrate you are providing, the reaction won't get reversed. And in this case, since the reaction is not getting reversed, Vmax would decrease. Means we cannot achieve the same maximum velocity. So Vmax would decrease while Km stays constant, as the affinity between the substrate and enzyme is not affected. So let's look at the next slide. Wait. As you can see, the enzyme, the inhibitor can bind to the enzyme at the first place, means where the at the place where the substrate is binding, as well as at the level of the enzyme substrate complex, at this site as well. So in the presence of a non-competitive inhibitor, the Michaelis-Menten equation gets modified and it somewhat looks like this. So we'll talk about this in detail in some other lesson, but this is not for NEET aspirants. So let's look at the other slide. This graph is again a very good indication of what is happening. On the x-axis you can see substrate concentration, on the y-axis there is maximum velocity, and as you can see, since the inhibitor is not competing with the substrate for binding with the active site of the enzyme, the Km value is same, since the affinity is not affected while the Vmax is decreased. Similar is being shown in this curve as well. This is Lineweaver-Burke plot. So as you can see, the Km is same while the Vmax is decreasing. The Vmax is decreasing. So let's go to the next slide. And this is the example of non-competitive inhibitor, cyanide poison. Cyanide poison binds to the copper ions present in the cytochrome oxidase enzyme. This is an enzyme which remains associated with the mitochondria. This is a transmembrane enzyme which remains associated with the mitochondrial membrane, and it is very important for the regulation of cellular respiration. So when cyanide poison binds with the copper ion present in the active site of this cytochrome oxidate, this enzyme stops, leading to the disruption of the cellular respiration and death. So this is the example of non-competitive inhibition. And now we will look at uncompetitive inhibition, and this part is not mentioned for the NEET aspirants in the NCERT books. Uncompetitive inhibition, when an enzyme inhibitor binds only to the enzyme substrate complex. As you can see, the inhibitor is binding only at this side, while in the first case, if you remember, in case of competitive inhibition, the inhibitor was binding at the first site where the substrate was binding. In the second case, where of non-competitive inhibition, you have seen that inhibitor was binding at this site as well as this site, and while in case of uncompetitive inhibition, it is also called as anti-competitive inhibition. The inhibitor is binding only at the enzyme substrate complex site. And uncompetitive inhibition typically occurs in a reaction with two or more substrates or products, and in this case as well the reaction gets modified slightly. Let's look at the next slide. So this is the curve which is indicating the uncompetitive inhibition. As you can see, the Km value is decreased here, means Km is decreased, as well as the Vmax is decreased here. So let's look at the example of this. Inhibition of acetylcholinesterase by tertiary amines and alkaline phosphatase operate via this mechanism in cancerous cells. And here is a short summary of what we have studied in different types of inhibitions. In competitive inhibition, the Vmax, there is no change in the Vmax, while the Km is increased. Km increased means the affinity of the substrate for the enzyme decreases because the inhibitor compete with the substrate for binding with the active site of the enzyme. In non-competitive inhibition, as you can see, the Vmax value is decreased as maximum velocity can't be achieved because the inhibitor can bind with the enzyme both at the when enzyme is alone as well as when enzyme is bound to the substrate. And in case of uncompetitive inhibitors, the Vmax decreases and Km decreases as well.